take one breath right now before you read another word. In through your nose for 4 seconds, out through your mouth for 8, and really stretch that exhale out. Now check your heart rate at the start of that breath and check it again at the end. It slowed down. And here's the thing, it didn't slow down because you relaxed or because your mind got quieter. A specific nerve, running all the way from your brainstem down into your gut, physically reached into the electrical wiring of your heart and pulled the brakes. That nerve is called the vagus, from the Latin word for wandering, and honestly, the name barely does it justice. It leaves the skull, drapes down both sides of your neck, sends branches into your larynx, your lungs, your heart, your stomach, your intestines, your spleen, and finally trails off somewhere near the transverse colon. It's the longest cranial nerve in the human body, and for most of the history of medicine, it got treated like a single wire doing one dull job: slow the heart down, help digestion along. That description isn't wrong, exactly. It's just missing almost everything that matters. Here's the number that should stop you cold. Somewhere between 80 and 90% of the fibers inside the vagus nerve aren't carrying instructions downward, from your brain to your organs. They're carrying information the other way, upward, from your organs to your brain. Stephen Porges, the neuroscientist at Indiana University, who spent decades mapping this system into what he eventually called polyvagal theory, put it about as plainly as it can be put. The vagus is primarily a sensory highway, not a motor one. Your gut is talking to your brain roughly 9 times more than your brain is talking back to it. The organ you've always pictured as silent background machinery is actually one of the most talkative structures in your entire body, and you're not conscious of almost any of that conversation. Let's start with the heart, because it's the layer you can actually feel. The sinoatrial node, your heart's natural pacemaker, sits in the wall of the right atrium and fires on its own. Left completely alone, with zero nervous input, it would drive your heart at roughly 100 beats a minute. The only reason your resting heart rate sits closer to 60 or 70 is the vagus nerve, constantly dripping acetylcholine onto that node, binding to muscarinic M2 receptors, popping open potassium channels called Gurk channels, and hyperpolarizing those pacemaker cells so they fire less often. This isn't some slow hormonal drip either. Acetylcholine gets broken down in milliseconds. The vagus can speed your heart up or slow it down within a single beat, which is exactly why your heart rate visibly climbs when you inhale and drops when you exhale. That's called respiratory sinus arrhythmia, and it is the precise mechanism you just felt in the breath you took 30 seconds ago. That one mechanism alone would already make the vagus one of the more important nerves you own. But it never works alone. Woven into the wall of your aortic arch and your carotid sinus are pressure-sensitive nerve endings called baroreceptors, and the signal they generate travels back to your brainstem mostly along vagal afferent fibers. The moment your blood pressure ticks up even slightly, a few millimeters of mercury, nothing dramatic, those stretch receptors start firing faster. Your brainstem responds by cranking up vagal output to the heart, and your heart rate drops within about 1 to 2 seconds to bring the pressure back down. That's the baroreflex. It's fast, and it's precise enough that anesthesiologists actually watch for it breaking down during surgery. When that reflex stops behaving normally, it's one of the clearest red flags that a patient's autonomic nervous system has come dangerously unmoored. Every time you stand up too fast and feel that half second of lightheadedness before your vision steadies out again, that's you feeling the brief lag before this exact vagal loop catches up to you. But there's a third mechanism running through this same nerve, and it was found almost by accident, in a hospital lab, not a physiology department. Back in the late 1990s, Kevin Tracey, a neurosurgeon at what's now the Feinstein Institutes for Medical Research in New York, was chasing a drug to block a lethal inflammatory molecule called TNF-alpha in sepsis patients. Almost as an afterthought, basically a control experiment, his lab electrically stimulated the vagus nerve in rats before injecting them with a bacterial toxin. And the inflammatory response, cytokine levels that should have spiked into lethal territory, dropped by nearly 75%. A single nerve, stimulated for a few minutes, had blunted a full-body immune reaction more effectively than most of the drugs available at the time. Tracey called it the cholinergic anti-inflammatory pathway, and over the following decade he chased the mechanism all the way down to the molecular level, publishing the core finding in Nature in the year 2000. Vagal signals travel down to the spleen, where they release acetylcholine near a population of T cells that turn around and produce even more acetylcholine themselves. That acetylcholine binds to something called the alpha-7 nicotinic acetylcholine receptor, which sits on macrophages, the very immune cells responsible for pumping out TNF-alpha in the first place. Flip that one receptor on, and cytokine production drops almost immediately. Your immune system, the thing most people picture as purely chemical, antibodies and cells drifting around in your blood, has a direct electrical off switch, and it's wired through the exact same nerve that just slowed your heart rate down. For most of the 20th century, immunology and neurology basically lived in separate buildings. The immune system was chemical, antibodies, cytokines, slow cascades of signaling molecules. The nervous system was electrical, voltage, ion channels, milliseconds. Tracey's discovery forced those two fields to collide, and the pushback inside immunology at the time was real. A claim that a few minutes of electrical stimulation could outperform pharmaceutical anti-inflammatories sounded, frankly, too clean to believe. It took roughly a decade of replication across multiple labs and multiple inflammatory models before the field accepted the pathway as genuinely real. And this isn't some fringe idea sitting in a journal somewhere. It made its way into human medicine, and earlier than most people would guess. Implantable vagus nerve stimulators, a small electrode coiled directly around the left vagus nerve in the neck, wired down to a pulse generator tucked under the collarbone, have been FDA-approved for treatment-resistant epilepsy since 1997, and later for treatment-resistant depression too. Epilepsy came first, and for years the mechanism sat inside a black box. Stimulate the nerve, seizure frequency drops in a meaningful chunk of patients. Nobody could say with total confidence why. And here's where the story takes a turn, because a completely separate line of research, out of Michael Kilgard's lab at the University of Texas at Dallas, stumbled onto something neither the sepsis researchers nor the epilepsy researchers had been looking for. Pairing brief, half-second bursts of vagus nerve stimulation with physical movement during stroke rehab dramatically sped up motor recovery in randomized trials, compared with identical movement therapy without the stimulation. The mechanism is neuroplasticity. Vagal stimulation triggers a release of norepinephrine and acetylcholine in the motor cortex at the exact moment a specific motor circuit is active, essentially tagging that circuit for reinforcement while it's still firing. Think of it as the nervous system highlighting a single line in a textbook the instant you read it, instead of hoping you'll remember which line mattered later on. In 2021, the FDA approved a paired VNS device for chronic stroke-related arm impairment, branded Vivastim, based on a randomized trial published in The Lancet. A nerve that was first mapped for digestion, then remapped for immunity, is now an approved clinical tool for teaching a damaged motor cortex how to physically rewire itself. Now think about the gut itself for a second, because this is the layer research is only just starting to map, and it's also the layer where you need to be a little more careful with the claims. Your intestines contain what's sometimes called the enteric nervous system, roughly 500 million neurons embedded right in the gut wall, enough that people occasionally, and a bit loosely, nickname it a second brain. Vagal afferent fibers end near this system without actually pushing through the intestinal wall, which means they can't sense bacteria directly. What they sense instead are the downstream chemical signals, short-chain fatty acids like butyrate and propionate, shifts in gut hormones like cholecystokinin and GLP-1, changes in local serotonin, over 90% of which, by the way, gets produced in your gut and not your brain, mostly by a specialized cell type called the enterochromaffin cell. Diego Bojorquez's lab at Duke University found a more direct route in 2018, a class of gut epithelial cells they named neuropods, which form actual synapse-like connections with individual vagal nerve endings, releasing glutamate across that junction in under 100 milliseconds. That's faster than most hormonal signaling by orders of magnitude. It means a subset of your gut cells aren't just dumping chemicals into your bloodstream and waiting around. They're firing directly onto a nerve ending, the same way one neuron fires onto another, giving your intestine a synaptic connection to your brainstem measured in milliseconds instead of minutes. A study out of McMaster University, led by Premisel Birchik and John Bienenstock, tested this pathway head-on. Mice fed the probiotic strain Lactobacillus rhamnosus showed measurable drops in anxiety-like behavior, along with altered expression of GABA receptors in the brain, the same receptor system targeted by drugs like benzodiazepines. Then the researchers surgically severed the vagus nerve in a separate group of mice before feeding them the same probiotic, and both the behavioral change and the receptor change vanished completely. The bacteria weren't acting through the bloodstream at all. The signal needed an intact nerve to travel through. And here's the evidence calibration, because this is exactly the kind of finding that gets flattened into overblown claims online. That result is solid, replicated, mechanistically precise in mice. In humans, the vagal gut-brain connection to mood is anatomically plausible and has some support from smaller trials, but it's nowhere near as settled. John Cryan and Ted Dinan at University College Cork, who actually coined the term psychobiotics for this whole research area, have said outright in their own papers that human microbiome to mood causation still needs far more rigorous trial design before anyone should be making clinical claims about it. The physics and the biochemistry of the pathway are proven. A confident claim that your yogurt is treating your anxiety through your vagus nerve is not. So here's what's actually happening all at once, all the time, without asking your permission or your attention. Your heart rate is being adjusted beat by beat through acetylcholine release at the sinoatrial node. Your spleen is receiving signals that dial down inflammatory cytokine production through the alpha-7 receptor pathway. Your gut is sending a nonstop stream of chemical and mechanical data upward. 9 parts sensory information for every 1 part motor command, describing stretch, pH, hormone levels, bacterial metabolites, all of it. Every one of these streams converges on a single brainstem structure called the nucleus tractus solitarius, a relay station roughly the size of a grain of rice, which pulls together cardiovascular, respiratory, immune, and digestive information before shipping it upward to the hypothalamus, the amygdala, and the insular cortex. That upward projection is what eventually becomes your felt sense of your own internal state. What researchers in this field call interoception, your brain's continuously updated model of your own body. A.D. Craig, whose work at the Barrow Neurological Institute helped build the modern map of this system, described interoception as the actual physiological basis of subjective feeling. Not as a metaphor, but as the literal neural substrate that emotional experience gets built out of, moment to moment. Under that framework, a feeling in your chest isn't standing in for anxiety. It is, in a very direct anatomical sense, your insular cortex reading vagal traffic in real time. This is the part of the story I find genuinely striking. Porges's polyvagal theory argues the vagus nerve doesn't just report calm back to you, it actually produces the physiological conditions that make calm, safety, and social connection possible in the first place through a specific branch called the ventral vagal complex, which also happens to innervate the muscles of the face and larynx. That links heart rate regulation directly to the exact muscles you use to make a warm expression or soften your voice. Now, whether that particular evolutionary framing accounts for the full complexity of human social behavior is genuinely debated among physiologists. Porges's broader theoretical claims have drawn real, scientific criticism for outrunning the data in places. But the underlying anatomy isn't in dispute. The same nerve regulating your heartbeat physically shares brainstem circuitry with the nerve controlling the muscles around your eyes and the pitch of your voice. Which brings us back to the breath you took at the very start of this. A slow exhale measurably increases vagal tone. Clinically, it gets quantified as heart rate variability, the beat-to-beat variation in the gap between your heartbeats. And higher variability is one of the most replicated biomarkers of vagal function in all of cardiology. Low heart rate variability, meanwhile, is independently linked to a higher risk of all-cause mortality in large longitudinal studies, including a frequently cited analysis out of the Framingham Heart Study. That 4-second inhale and 8-second exhale you just did wasn't some wellness technique borrowed from folklore. It was a mechanical lever, pulling on the exact same acetylcholine pathway that Kevin Tracey used to blunt lethal inflammation in a rat, and that Michael Kilgard's implanted electrodes are using right now to help stroke patients relearn how to move their own hands. You can't feel any of this happening. Not the potassium channels popping open in your sinoatrial node. Not the baroreceptors in your aortic arch correcting your blood pressure one heartbeat at a time. Not the acetylcholine reaching your spleen. Not the neuropods in your intestinal lining firing glutamate onto a nerve ending in under 1/10 of a second. The entire system runs below the threshold of conscious awareness, and it's been running that way since before you had language to describe any of it. Adjusting your heart, your immune system, and your gut in a continuous, wordless negotiation you were never once invited to consciously join. There's one exception, though, and it's a narrow one. Breath is the single rhythm in this whole network sitting right on the border between automatic and voluntary. The only vagal input you can actually reach in and operate by hand, the way you can't manually operate your spleen or your baroreflex. That's not a wellness trick bolted onto the physiology afterward. It is the physiology itself. It's the one door in the entire system that was ever left unlocked. You were only ever given the exhale. That was the door they left open.